Sonnenberg, and they have been working on the project called Chickadees, research project on chickadees under this professor, and who I believe is world renowned for studying chickadees. So they're going to share some of their research with us, and we're very excited to have them. I don't know which one's going first. Ben Sonnenberg. All right, please help me welcome Ben.
Yeah, that's right. Okay, can somebody put the PowerPoint up for me?
Sure can.
Hello. Apologies that you get me first. So that's the bad news. The good news is you get Lauren last. So
So Ben, when we get this all going,
yeah,
you have to stand in front of the camera.
so that it will be
If you need us to turn it to see something, we'll do that.
sure.
Yeah.
I'm a wanderer, so no promises for a
Start.
continuous stream of my under chin. Okay, great. Yeah, so my name is Ben Sonnenberg. I'm a PhD candidate at the University of Nevada, Reno. And both Lauren and I are part of the Ecology, Evolution, and Conservation Biology program there. And I see a lot of familiar faces. And so I'm sorry if you've heard, some of the, the wonderful chickadee work before, but we'll just get to talk about it again tonight. Maybe. Okay, we'll do that.
Yeah, you said the click.
Okay, I can't. Yeah, there we go. Perfect. Okay, great. So I'm a behavioral ecologist, and essentially we are looking at chickadee behavior, and we're particularly really interested in their caching behavior in our lab. So there are several behavioral ecology labs at the University of Nevada, Reno. If you want to visit the website of our own lab, that's chickadeecognition.com. So if you guys are hankering to visit. And then Lauren and I also have websites of our own that we can share if you guys are interested in looking at our specific work and some of the things that we do on our own. But so today I'm going to be talking a little bit about cognition and the cognition that's behind the behavior that we study, which is food caching. And food caching is essentially hiding food in the woods, right? So squirrels, right, put food, right, and sometimes large collections, or like acorn woodpeckers, like one that showed up in Reno is another example of an animal that puts a bunch of food in one place. Those are called larder hoarders. And so it's essentially what we do. We're larder hoarders as well, right? So we actually keep a fridge, right? How many of you guys have a fridge, pantry? You guys are all larder hoarders and didn't know. Well, the bad news is that larder hoarders in general have very poor spatial memory ability. And explains a lot, right? And so if you guys have ever lost your car keys, right? Chickadees have never lost their car keys because they are scatter hoarders and have evolved a unique number of traits, including brain morphology and behavior that goes along with that brain morphology that allows them to remember. Does anybody want to guess how many individual seeds that chickadees cache in individual locations every fall and winter? I heard in the back. Yeah, tens of thousands. So individuals can cache up to 100,000 individual seeds. And these guys are not seed dispersers either. We have a world-renowned memologist here in the room too that has worked on a lot of food caching, well, some food caching rodents, right? But not in maybe the capacity of food caching. You've done food caching work, yeah? Yeah, absolutely. So then they're not actually, these guys, our chickadees are stingy. They're not good like the rodents. In fact, they cache their seeds way up in the trees and underneath like pine needles and in between bark. So they do not actually plant their seeds, right? So if they forget about a cache, it actually just, essentially doesn't actually result in a seed. But so we're focused on cognition, and I'm going to be saying that probably many times tonight, but that definition is the mechanisms by which animals acquire, so learn, right, process, store, and act on information from their environment, right? And so this includes perception, right, learning, memory, and decision-making. And so our lab primarily focuses on learning, and specifically spatial learning. And so we study this adorable, some would say maybe the best bird in the world. Some would say that. the mountain chickadee, right, which is a resident to Western North America, and really primarily they only are in coniferous forests. So we study them by where this yellow star is marked, so which is Sept Experimental Forest, which is around 15 miles north of Truckee on Highway 89. And chickadees are really wonderful. So they have a bunch of really interesting traits that make them lovely to study. And I'll be talking about more in depth, and Lauren will be talking about some of their care and some of the things that we monitor. But they exhibit biparental care. So that means that both parents care for the young. They're relatively short-lived, but they're highly resident. And so our chickadees, once we ban them, we see the same individual. And we have a couple 9-year-old birds in our system, as well as multiple 6-year-old birds, et cetera, et cetera. So we can track these guys across many, many years. And then, of course, they have specialized spatial cognitive abilities that will allow them to cache food. And so Lauren and I are part of what's growing and growing every year, but it's a long-term spatial cognitive monitoring study. And so this is the only one of its kind in North America, and it's actually pretty unique, on the world level. So we monitor chickadees, we ban chickadees in the wintertime, and then we monitor their cognitive abilities in the wintertime. Then we track those same individuals into the reproductive season, and we monitor their reproductive output. And just to give you some, and it's terrible. No one has any fun. It's the worst field work in the world. No, it's amazing. We get to have a lot of fun. So we have really specialized equipment to be able to work throughout the winters, right? Monitoring some of these guys. Cognitive abilities, and it takes a lot to maintain this long-term system, right? So this has been going on for 11 years now. We have banded over 2,400 adult chickadees with passive integrated transponder tags, which is what I'll be talking about in a minute. But those are unique individuals, 2,400. We've tracked over 1,100 nests and banded 5,200 chickadee nestlings. And then of all these individuals, we've measured the cognitive performance of over 750 individuals. And that is the most in the world, right? We've done a lot with monitoring individuals, not only their reproductive output, but connecting it to their cognitive ability. So some of the comparative studies, if you've heard of systems like Wytham Woods in the UK that monitors great tits, they're going longer for us with monitoring reproductive output, but we are doing a little bit more than that, which is really fun. So our field site is situated across a montane elevation gradient. So you might be hearing us refer to low elevation or high elevation in this talk. And that's, again, back to the fact that these chickadees are really highly resident species, and they cache because they don't move, right? So when they're living at the very tippy top of the mountain, say at 8,300 feet, right, they're going to stay there throughout the entire winter. They don't move. They don't fly down. They don't come to more luxurious place, because that would be weird, right? You're leaving your fridge at the top of the mountain. You've already made your fridge. You've put the things in the fridge. You can't leave your fridge, right? And they don't need to leave because they have that food resource. And so our high elevation site is around 2,300 meters. Our low elevation site is around a little over 1,900 meters. And I have that center line in there because it's why we study them across that elevation, is because those two elevation points are situated across what's called a snow line, which you might have heard a weatherman mention, right? But the snow line is actually an approximate elevation that changes year to year and situation to situation. But in our system, it's around 2,100 meters. That during transitional months of the year, such as spring and fall, precipitation above the snow line at higher elevations is more likely to fall as snow. And below that snow line at lower elevations, precipitation is much more likely to fall as rain, which translates to the higher elevation birds and individuals experiencing harsher winters and more snow, prolonged colder temperatures for longer. And so there's actually a difference in the need of caching, right? So if you're more cold, you have a longer winter, you need to cache more food, right? And so we actually see those differences in our birds. And we know that spatial cognitive ability, so the ability to remember more locations where you hid your food, is selected for at high elevation, even stronger than low elevations, right? So remembering where your food is really important and critical to the survival of these animals, especially at high elevation. And we use that comparison to compare a lot of different things about their biology. So some of you guys have seen this before. So I mentioned passive integrated transponders. That's this little white tag right here. But these are an example of the unique color combinations that we tag chickadees with. And that passive integrated transponder acts like a key. And so we have these really fancy radio frequency identification equipped feeders. And yeah, I forgot to mention, guys, there'll be a quiz at the end of this. So acronyms are not accepted either. So RFID, yeah, anyway, and spelling also takes. So it actually acts as a key that allows them access to our feeder systems. And so this bird is going to fly up and land on our feeder, and you can see that door open in the perch, and he's going to pick a seed and fly away, and then that door closes again.
Is it opening in response to that?
So I brought one of our feeders today, right? And so we're gonna be chickadees. And so we have this feeder, right? And I have an example transponder. So if a bird lands on the feeder, do you see our door open and close? And so then that actually allows us to measure the timing of the visitation and how many visits per day. So right now we actually are seeing some individual feeders are getting over 3,000 visits a day in our system here in September. And that's because birds are caching right now. So they're hiding food, right? So they're visiting a lot and they're hiding some of those sunflower seeds away. How's it going?
Oh, it decided it wanted to disappear. Hold on.
So Ben, 3,000 visits.
Yeah.
How many birds do you think that is?
You know, I actually, we did know that. It's around like, it varies by feeder. So we have these individual feeders that is measuring visitation rates. And it's like 40 to 50 birds or something like that per feeder. But we have overlap between feeders because some of them are close by, right? But right now we have 120 or 121 or something like that individuals visiting feeders at our high elevation site, and around 95 individual birds visiting at low elevation. Of course, that doesn't include birds that are not tagged, right? Which I'm sure we're getting. Yeah, okay.
We'll give you a chance to check on that.
Yes, sir. Yes, Dr. Sir. So with these particular feeders, right, we're able to measure two different cognitive tasks. So a spatial learning task, which measures their ability to remember a location in space, aka how well do they remember where they cache a seed, right? As well as a single spatial reversal learning task. And so then that's essentially their flexibility with learning, right? So how well are they able to make a new memory, essentially, right? And so for a lot of these animals, caching in very similar locations can actually be almost hazardous to the memory, because what you have happen is proactive interference, it's called, with two memories interfering with each other. And so for some of the birds that are living at high elevation, where it's really important to have memory and to remember exactly where that cache is, you have really strong memories that can interfere with the other. And so we measure really unique aspects about their abilities in that way too. And so to give an example of proactive interference, how many of you guys have ever had to get a new phone number in your life? Yeah. So it's much harder to learn a new phone number that is one digit off from your old one compared to learn a phone number that is completely different from your old one, because the similarity of that new phone number to your old one interferes with that other memory. So that's what I'm talking about when proactive interference. And so what these memory tasks actually look like in the wild is we have 8 feeders that hang up in this spatial array. At first, we habituate all the birds, so all the feeders open for them, but then we assign individuals only one location that provides a reward. And then we see how well that individual learns that. And so for what the task might look like initially is that a bird might come in, he might look around, oh, well, that feeder used to give me seed, no more. So then he looks around and samples his second feeder, oh, still no seed. So then he samples the last feeder and finds his rewarding location, yay. So he might fly off, he might cache that seed, he might eat that seed. But what happens when he comes back is really important, right? So he came back and he went straight to that that rewarding feeder, right? So we can measure that he learned, right? He got a question right on the test, right? And we can measure how fast they learn by how many errors they make per trial. So each time they come to the feeder, so each one of these is the time they come to the feeder, they might sample 3 feeders before they find their reward, then 2, and then they might never make a mistake again. So you can see that's a learning curve, right? They're improving over time, right? So we can measure their learning speed and then just their learning ability. So let's test your memory. I'm going to give you 10 seconds to memorize where these different animals are on this slide. 5 or 10, 9, 8, 7, 6, 5, 4, 3, 2, and then we're going to cover them all up. So now we're going to ask you, where was the dinosaur? Number 3. Ding, ding, ding. Awesome, right? So but that's an example of a spatial memory test that we'd give to humans. And I have a harder one, but we don't have time tonight. Right? So you made 0 errors, right? And so for our chickadees, over time, we measure their memory ability as well. And does anybody have any guesses on their average performance on this task when it comes to error? So I will say too that we have 8 feeders, right? And so if they visit 3 or more feeders, it's probably they're sampling from random chance and they're not learning anything, right? But if they make anything below like 3.5 errors, so like 2 errors or 1 error, that means that they're learning something. And so what do you think? How do you think chickadees do? What is it? 0?
30%.
30%? Okay, yeah.
High elevation or low elevation?
Let's do both. Yeah, we combine both today. Okay, so I have a slide with both juveniles and adults here, but you can see they make on average, under an error. And so a lot of the times, the reason why we see any error, right, is because they have to initially sample their feeders, right, and find which one is theirs. But then after they find their initial one, they never make a mistake again. And so for other species, when we look at some of the other data and other people trying to do cognitive work in the wild, a lot of animals simply do not learn the task, right, at all. And a lot of them have terrible performance. We have people come up to us at conferences all the time and say, why are your chickadees so smart? You need to make the task harder. They're clearly, but it's actually because of their amazing natural history, right? They're actually caching. They need this memory to survive. And that's why they're so good at this task. And that's why we're able to see such really cool performance on this task. But it's actually to do with their natural history. And so we track these things across all those years, right? So I said that we had measured over 700, right, individuals on the spatial memory abilities, right? Across these 11 years, we've measured cognition for 8, and we've measured, over 1,000 nests. And what we look for when we monitor reproduction is we look for things like nest building behavior, which is something that I'm intimately familiar with and has been part of my dissertation. I'm really particularly interested in how early developmental environments within the chickadee nest and just surrounding their biology affects their future cognitive ability, right? Cognitive ability takes a lot of brain power. And if you're not being fed the right things, if you're not being fed well enough, right, it can result in just poor development and poor spatial cognitive ability. We also measured the date of first egg, which I'll let Lauren tell you about why that's so important. We also look at hatch date. So these are 2 little tiny chickadees that are less than 12 hours old. And that's the male coming in probably to feed not just the young, but to feed the female as well so that she can stay in the nest box and keep those nestlings warm as the rest of the clutch hatches out of their egg. So they're really good parents. And then we also look at the nestlings. And so this adorable little fellow is only 16 days old. So they grow quite fast. And we do lots of things on day 16. We take a DNA sample so we can look at who they're related to. We also take measurements such as mass, because that can tell us about the health and quality of the nest. And then we also look at things like the time of fledging, so when these guys are actually leaving the nest. And so, like I said, we've done a lot of work on all this, and we've are just starting to get to some really cool results within our system. But like I said, I'm really interested in some of the aspects shaping parental care. And so some of my projects don't encompass the full length of our data set, right? So I've been here for 6 years and have contributed to 6 years of data collection within the system, which is awesome. And I've learned a lot throughout this time, but I'm really interested in parental care and some of the differences that we might be seeing in the offspring that's related to maybe where they're living, right? So environmental conditions such as harsh winters, right? So those differences between high and low elevation. So how do parents might differ between high and low elevations? The social structure, right, of parents also might help, so that the fact that they're monogamous, right, how does a male feed more than a female? So the social structure and the communication between social pairs at the nest box might be really important to development. The life history strategies, we actually have a reason to believe that chickadees between our low and high elevation sites might be separating a little bit and might be having different strategies. And individual quality of the parents, right, can all influence things like, how many offspring are produced in a nest, right? And so one of the projects that I did to look whether or not this was the case was I wanted to see how attendance at the nest box, so how many feeding events and how they might differ between an experienced pair or an inexperienced pair. So whether or not a pair had raised nestlings before, whether or not they had never raised a clutch before, as well as the composition of the pair, right? So does a male feed more? Does a female feed more? Does it matter within the nest time? And does that result in better nestlings, right? With better future spatial cognition? And then I also just wanted to know whether or not birds with better cognition fed more, right? Did they have the capacity to feed more offspring to their young, right? Because if you have more food hidden in the woods, you might come out of winter in much better condition. And so you might be able to feed, right? And you may be able to produce more nestlings. And so I took advantage of all these birds with those RFID tags on them, like this one, and we'll pass these guys around too. And we actually put antennas, that's what this is right here, an antenna in the side of the nest box, and we monitored how many times mom and dad went out of the nest. And we can do that because we have little tiny microcomputers that essentially fit inside Tupperware with batteries strapped to the back of a tree, really high tech, to measure some of that stuff. And so what a parental visit looks like, so here comes the male chickadee. So there's his pit tag, and you can see he's got a good number of food items in his bill. So he climbs into the box and he's red. So we can actually measure how much time he spends down in the box feeding his nestlings. And then he's gonna pop back out, fly away. And we can measure that as one visit, right? And then we can see how many times these birds are visiting their nests and what that actually means. And so this is a giant bowl of colorful spaghetti. I'm sorry, this is a very serious scientific diagram. So this is actually on the y-axis here is the number of visits. And so you can see that some animals actually visit their nests over 1,000 times per day, right? So this is the cross, this is days in the nest, right? So this is day 1 all the way to day 22. And you can see when these birds fledge too, right? Do you see the drop off in the visitation? That's obviously when the babies are leaving the nest. And you can see the visitation starts out slow and grows and kind of spikes here in the middle and then declines. And that's kind of what we expected. But why do we see so much variation, right? You can see all that variation. And does that variation mean anything? Does that relate to some of these things that we thought? So we thought that maybe it would vary by year or by our elevation site. We thought that maybe it would vary by experienced pairs, right? So these birds that have more or less experience. And we also thought that individuals might visit differently depending on their cognitive abilities. And so if you look at some of these, so now we're looking at nest attendance, so number of visits per day per nestling. And you can see that each parent is visiting the nest over 30 times per day per nestling. But then it starts out slow, and then increases, and it peaks in mid-development, and then it declines again in late development. And that was all the same across years or in elevation. We didn't find any effect of year or elevation. And then we didn't actually find any effect of parental ability either, which I'll talk about in a sec. The only thing that we did find was that males and females, so this effect was driven by both parents. So if parents were feeding more early in development, that meant that their babies actually had higher mass when they were ready to fledge. And so nest attendance earlier is really important. So that's one thing that we did figure out. But we, like I said, didn't find any relationship between performance on our cognitive tasks. So whether or not they had superb memory ability or really poor memory ability, that had no impact on how good of parents they were. Then we also found no difference in attendance between parents with prior breeding experience. This is kind of odd because we have this tremendously large data set, and we've actually detected differences in nestling quality with parents that have prior experience. So birds that have bred before in our system actually normally produce more babies and higher quality, bigger babies. And so the fact that we didn't see a relationship between nest attendance, whether or not these parents are actually investing more, visiting their babies, was kind of interesting. And we didn't really know why that was the case. And that actually leads to the segue. Do you have a question?
I was wondering, are they mostly feeding their cached seeds, but aren't they also looking for things like spiders and, you know?
That's a super good question. Yeah, and I didn't mention that earlier on. So these guys are only eating seeds in the fall and the winter. And so then in the summertime, they switch entirely over to insects. And so they would be consuming insects, and then they would also be feeding insects to their young.
So they're bringing mostly insects when you're measuring the breeding.
Only insects. Yeah, only insects. Yeah, no seeds. So the only exception, which are amazing critters, that actually feed caches that they make to their young, are Canada jays. And the other two species of related jay that live over in Europe and Asia. And they make these really delicious bowls, or they're called boluses, which is just, a round ball that's covered in their saliva, which actually has antimicrobial properties. And it's like a stick of gum. So they make this cache of insects and then they stick it on things for later. And they will come back and feed their nestlings those caches. Our chickadees don't feed any of their nestlings caches. So that caching is a pure individual endeavor, right? And it has to do with those, individual quality of the chickadees once they come out of winter, but they're not actually feeding any of their babies, right? Seeds. But we didn't really know what they were feeding, right? And we didn't know how many insects were out there. This was like a gaping hole in our system that people asked us about all the time. And frankly, it was annoying. Because we didn't have anybody with the expertise or crazy wild ambition to go, I don't know, beat a bunch of trees for months of the year to actually sample the insect diversity that's out there. And so we actually came away with the idea that it's likely, because we do know that there is differences, especially in experienced pairs in reproductive performance, but not related to their visit number, but it's probably the quality of the visits that matters, right? Not the total number, but the quality, AKA what they're bringing, and the actual quality of their visits. And now I'm going to turn it over to Lauren. Yeah.
I just wondered if the bad winter that we had this last year affects this behavior. It must.
Yeah, so essentially what we see in the broader population with really bad winters is we see just higher selection, right? So more animals die in harsh winters. And just like some of the animals with the best cognitive ability stick around. And so this last winter was tough, but we have plenty of animals survive. The toughest part, or one of the toughest parts, especially at our high elevation site, is Lauren will show some pictures here in a second, is that we still had, 6 feet of snow in June, right? And so these animals rely on cavity nests, and we were digging our nest boxes out of 3 feet of snow in order to raise them up on the tree high enough that chickadees could access the boxes. And so that's a huge limitation. So that is tough on the birds, and some birds either skip reproduction or aren't able to reproduce at all in seasons like that. Yeah, okay.
Yeah. All right. Transition slide. Hi, everyone. My name is Lauren. I've met many of you on walks. I'm a board member of the Lahontan Audubon Society, and also I lead a lot of field trips. But I also, on the side, am a grad student. I'm also a PhD student in Vladimir Pravosudov's lab with Ben. And yeah, so I'm interested in how animals respond to environmental change and how trophic systems, so animals that interact with each other, respond to environmental change. And luckily, when I joined the lab, we had already collected our 10th year of breeding data. And so I had this amazing long-term system to work with in order to start asking some of these questions. And so just to go over really quickly the breeding data that we collect every year, we start monitoring these birds in April, and we record when they're nest building, when they lay their eggs, how many eggs they have, the hatch date, and then eventually on day 16, we grab all the nestlings and put little bands on them, we weigh them, and we count the number of nestlings that survived to that age. And so just jumping back, why do we study reproduction? Why is it important to monitor birds like this over long periods of time? Well, I'll just go over a few of the important reasons. First, by studying these patterns, we can understand how populations are changing. So some years birds might have way fewer nestlings, and it's important to understand those patterns and then maybe to connect them to, well, why is this happening? So that goes along with my second point, which is understanding how the population responds to different environmental and ecological conditions. And then if we have this amazing long-term system where we actually monitor individual birds over their lifetimes, we can start to get at things like, well, how many babies did this bird produce in its entire life? And is there something about this individual bird that makes it more likely to survive and have more nestlings than other birds in the population. And then we can start getting at some questions about natural selection and things like that. Okay, so over the 11 years that we've now collected our breeding data, you can see that this is first egg date. So this is breeding timing, and it's day 1 is January 1, right? So this is around May or June. And you can see that there's a lot of variation between years and also between elevations. So here on the left side, we have our low elevation site, and this is years 2013 all the way to 2023. And then over on the right, we have our high elevation site, and you can see there's actually a lot more variation in the high elevation site. Does everyone see that? Does anyone have an idea why? Yeah, okay. Snow, right? And so at high elevation in 2017 and in 2019 and 2023, these were all really high snow years, and the birds bred a lot later than these other years. So we have this other component of climate, not just temperature, but also snow, that can influence timing of reproduction in this system, which is really interesting. We also see a lot of variation in mean nestling mass. So why do we measure mean nestling mass? What does this mean about individual chicks? Does anyone know? What would a really lightweight nestling? Yes, sickly, yes. So really lightweight nestlings probably were fed much less by the parents. They're probably less likely to survive. They may never even fledge. And when they do fledge, they are going to be lower quality than the rest of their siblings that are out there in the world. And right after the birds fledge, that's a really, really critical time. Juvenile survival in that stage is really low. And so birds need all of the fuel that they can get to survive that, right? And so mean nestling mass is an important measure of nestling quality and how well parents are doing at raising their chicks year to year. And so as you can see, we also have a lot of variation in nestling mass. And so I came in and I wanted to ask the question, what causes this variation? Is it environment? Is it ecology? Is it demography? Is it something else? And a really fortunate part of working in this system is that we have access to highly localized weather data located at both our high and low elevation sites, like right at the site. And so these stations measure precipitation and temperature, and they even take soil metrics. And so we can use these data to understand environmental patterns across years and across elevations at our site. And so these are some fancy graphs showing variation in daily temperature across years and across, so each of these different colored lines is a different year. And then across the x-axis, we have September through August. So this is basically within the annual cycle. How does the temperature vary? So you can see in the winter, the temperature is low, and then it creeps back up into the spring and summer. But there is variation year to year in temperature. And anytime in this cycle, this can affect these birds because they're year-round residents, right? And so both winter and summer, fall temperature can potentially be affecting these birds. So not only do we see variation in temperature, but we also see huge variation in snow depth. So low elevation site is on the left and our high elevation site is on the right. And you can see that snow depth usually peaks around March. And we have some years that are like incredibly higher snow levels than others, right? Especially at our high elevation site. Okay, so what does this mean in terms of explaining this variation in reproduction? So in other systems, like the great tit system, this is a great tit. They live in Europe. This long-term system that has studied great tits found that with climate change, they were shifting their breeding timing to be earlier. And they were shifting it to be earlier to match warming spring temperatures. And so this graph sort of shows this is like the sum of temperatures over a window of time in the spring. With increasing spring temperature, lay date decreases, lay date gets earlier, becomes earlier in the year. And they found that this actually corresponded really well with how caterpillars were shifting their timing in this system. And caterpillars are their main food source, right? And so I wanted to ask the question, how is climatic variation associated with differences in reproductive timing and success in our system? So are our birds also responding to spring temperature? Are our birds responding to snow depth? And then how are these affecting all of their breeding metrics, their breeding timing, clutch size, mean mass, et cetera? And so just really briefly, because this isn't the main part of my talk, we actually did not find that spring temperature caused our birds to shift. So something else is happening in our system. Over these 10 years, our birds did not shift their breeding timing with spring temperature. We did find, like we would expect, that our high elevation birds bred later in heavy snow years, and they bred earlier in light snow years. And then we found some other weird correlations, like, well, this one's a little less weird, but brood size at low elevation was reduced when it was a drier year. And so this could mean that the growing season was shorter, there were less insects out, et cetera, et cetera. We found some correlations with like fall temperature and things like that were a little bit less explainable. But overall, there was still a lot of variation that we couldn't account for with this analysis. And so this leads me to the bugs, the bug part of my talk, right? So there's this huge missing piece in our system where we have all this variation in these breeding metrics, but we have no idea what food availability looks like across these years. And we know that many birds rely on insects for food, particularly during the breeding season. And that these insects often show a distinct peak in abundance with time. So within a year, there's often a peak in insect abundance. And so theoretically, birds should be trying to breed so that they can match this window of time when there's the most insects out, so that they have a lot of food to feed their young. But insects are really heavily regulated by environmental variables such as temperature. So insect emergence is highly controlled by temperature. And then insects are also highly dependent on moisture too. And so birds can use environmental cues that are associated with insect emergence in order to time their breeding to try to match this ideal timing. But with climate change, these environmental cues that birds would use are shifting. So there's warming temperatures, but there's also increases in the frequency and the intensity of precipitation extremes, like this heavy snow, heavy drought cycle. And the worry is that with climate change, different animals will respond differently to these changes. And so in this talk, I'm going to use the term phenology. And when I use the term phenology, I'm just talking about the timing of seasonal ecological events, right? So phenological responses just means shifts in timing. So this graph shows that plants in green, invertebrates in orange, and vertebrates in blue in different systems, we have marine, freshwater, and terrestrial systems, show different magnitudes of response to warming. So they shift their timing to different degrees from each other. And these different levels rely on each other for food. They're connected, right? And so if these different trophic levels have different responses, this could lead to mismatches where, for example, birds no longer match the timing when there are the most insects about. And so this might lead to decreased reproductive success. Is everyone following me? Okay, please let me know if you have any questions. Feel free to raise your hand or yell at me. Okay, so some of this work has been done in low elevation deciduous forests, particularly in European systems like the Great Tit System, where they've looked at food availability by collecting caterpillar poop, because caterpillars are the main food source there. But really little is known about what's happening in coniferous forests in terms of what does insect abundance look like seasonally? How does this change with environment? And how are the birds responding? Very little of that is known. And well, voila, our system provides the perfect opportunity to look into these things. So mountain chickadees, like Ben said, they consume only seeds, only cache seeds in the winter. Maybe a few spiders or moths if they're around, but insect activity drastically declines in the winter, right? Picks back up in the spring, and then our birds completely shift their diet to insects. And so they're feeding their young insects, and they're also consuming insects themselves. And so I wanted to ask the questions, are chickadees matching this optimal peak in food availability? And does it matter for the reproductive success if they do or don't? So if birds better match this peak in abundance, do they have, let's say, nestlings with greater mass, or do they have more nestlings, right? And in order to do this, I need insect data. So I went out and followed this heavily, very detailed diagram, and collected insect data myself using beat sheets. So this is an upside down umbrella, but beat sheets are basically any fabric with a frame held underneath a tree branch. You hit the tree branch, and anything that is living on that tree branch falls onto the sheet, right? And then I count everything. So this is the main way that I count insects, and this is because our birds are primarily foraging on conifers. So they're picking bugs off of trees. But I also use malaise traps because beat sheets are undersampling flying arthropods, and malaise traps are these big hanging traps that passively collect flying insects. I also need to understand what our nestlings are eating so that I know if I'm measuring the right insects in the environment. And so luckily we have this, this amazing new technology where we can just collect their feces, extract the DNA, amplify it, sequence the DNA, and then match these sequences to existing libraries where we can identify insects even to species, the insects that they've eaten, right? So we do this.
So you're getting the food from the baby.
Mm-hmm.
You're analyzing that to say what they're eating.
Yep.
Amazing.
Yeah. So we no longer have to do things like dissect the stomach, because then you kill the baby, or right, or choke the nestling and prevent them from swallowing to collect the, right? Those were the methods that people used before. So now we have these really nice methods that are much less invasive. Okay, so I'm just going to present some data, hopefully relatively quickly. So this is 2022. So this is not this year, this is last year. We saw, based on this DNA analysis, that our birds were eating an incredible diversity of insects. So it is not all about caterpillars, basically, right? They're eating 25 orders of arthropods from 150 families. Some of the most common diet items were this guy right here, which is a type of scarab beetle that forage on conifers. But all of these photos I took either in the field doing my sampling or under the microscope when I brought the samples back home. And all of these are families that are represented in the diet. So we are sampling the right things, right? Okay, so now let's look at what the trends of arthropod or insect abundance look like over time in 2022. So I'm going to use this icon up here to represent beat sheet data, because I also have the malaise data, so there's a lot of data happening. But as you can see, this is date from April to October, and this is total biomass, which is estimated based on these regressions. Basically, I estimate the length of each individual specimen that I find, and then I calculate the biomass from that. And you can see that the biomass peaked in July at low elevation. It also peaked at the same time at high elevation. This is super weird because people that look at elevation gradients, normally phenology is later at higher elevations, right? Even our birds breed later at high elevation. So I think the reason this is because the peak is made up of different arthropod groups across elevations. Amazingly, our malaise trap data showed the exact same peak date, which was really cool. So how does this line up with when our birds are breeding? Well, each of these black bars shows a nesting period, meaning hatch day to when they're fledging. And you can see that this is our low elevation data set. our birds are all breeding before the peak. So like, what is happening with that, right? They're not matching the peak in food abundance. Same with our high elevation site. Our high elevation site matches a little better to the peak, is a little bit shifted because the peak occurred at the same time, but our birds are breeding later. But again, they're still breeding early. So do chickadees match the peak in food abundance? Kind of, no, right? Low elevation bred before the peak, high elevation overlapped a little bit with the beginning of the peak, but it doesn't seem to look that way, right? Well, now I wanted to ask, does it matter? And in order to do that, I calculated what we call synchrony, which is basically the difference between when the peak nestling food demand occurs, which we think is around day 12 in the nest. It's the middle period of the nesting period, like when Ben was talking, that's when the parents feed the most, visit the most. So the difference between that date and the peak biomass date. And this will sort of tell us how synchronized our birds are with the insect availability. And what we expected is that birds that bred farther from the peak would do worse than birds that bred closer. And this would translate to this sort of curved quadratic relationship between synchrony and brood size, where if birds perfectly match the peak, then they would do the best, right? But I will tell you now, and you've already seen those graphs before, that we did not see that relationship. We only saw a weird quadratic relationship between synchrony and mean nestling mass at high, but synchrony is not centered around 0. This quadratic relationship is not centered around 0. And we know that the experience of parents can influence how well the nest does. So we added parental age into the models. And when we separate it out by female age, so we have females that are breeding for the first time versus females that are older, we see that females that are breeding for the first time, a lot of them bred late and did really poorly. So we think that it's something to do with the quality of these females. After they come out of their first winter, they may be really struggling and they might delay their breeding and then also do really poorly or something like that. But we don't think that that relationship was due to food availability. So did it matter in 2022? It doesn't seem to follow the pattern of many of these other studies that have done this similar work. But that was 1 year, and I have, 10, 20, 30 more years of my PhD. But yeah, so interannual variation is going to be really, really important, I think, in this system. So in 2022, we had a sort of normal, sort of maybe low snow year. But in 2023, everyone knows we had a crazy snow year. Crazy, by crazy, I mean, literally in July, high elevation still had 3 feet of snow. And it looked like this. So we were walking that, and it was extremely difficult to walk in. And so I'm still collecting these data. I'm going to collect them until the snow starts. But this is what our data looks like from this year. So the solid line is 2023's data, and the dashed line is last year's data. And so our low elevation site, literally the peak was on the exact same day. I cannot make this up. It was crazy. But then biomass continued to be high much later in the season. And we think this is because there was much more moisture at low elevation this year for longer into the summer. And so this could have extended the growing season and extended, the insect abundance peak, basically. But high elevation was super, super interesting because entire families of insects were completely missing from my data. I have no idea what happened to them this year. They just never came out. So we had way, way, way lower biomass overall, and the peak occurred way later this year than last year. Yeah. Did you have a question?
So does that mean that the high elevation birds just weren't very successful this year?
I'm going to get there. But okay, yeah. So this is basically the same graph as before, right? Showing the nesting periods overlaid with food availability. And high elevation is really, really, really missing the peak, obviously. But so this is a graph showing brood size over all of the years of our data collection. And low elevation is in purple, high elevation is in orange. See that last year? they had really, really low brood sizes this year. And so I'm still analyzing the data, and we need many more years of data for this, but that definitely is an indicator that they were struggling this year, right? I also found this year that at low elevation, birds that bred later had nestlings with higher mean mass. So potentially birds that were matching the peak better, right? Closer to the peak, which obviously none of them were meeting the peak, but those that were closer did a little bit better potentially. Okay, sorry, this went a little long, but future directions, I need more bugs. I need more of them. I only collected 25,000 individual specimens last year and 40,000 this year, so I don't have enough of them.
I'm asking for volunteers.
Do you want to volunteer?
We have a room full here.
Yeah, so I'm going to collect more bugs, and I really want to look into all of these relationships between climate, food availability, and reproduction. I want to look at individual responses to environmental variation. So how do individual birds, how are they responding over time to different environmental variation? And then I also want to eventually forecast how, especially our high elevation population, would change under extreme climate change scenarios. So if climate were to change such that we have even more increasingly really, really heavy snow years and really, really dry years right in a row, or we have multiple heavy snow years in a row, and they have really, really small brood sizes all of those years, is their population going to be able to recover from that? So that's just one of the scenarios that I'm interested in looking at. Yeah.
I have a question with your beat sheets.
Yeah.
You're obviously sampling low in the
Yeah.
tree.
Yeah.
But the tree's a lot taller.
Yeah.
So do you think maybe the ones that are up at the top might be warmer and have more insects? And how could you even sample?
Yeah, no, this is a really good question. And this is one that a lot of people ask. And you're totally right. They're foraging at all levels of the tree. So we see them at the bottom of the tree and we see them at the top. We even see them sometimes foraging on the ground. So there is some, diversity that I'm not capturing with my sampling methods that could be relevant.
And I don't know the other one. Can they hang that big thing way up?
Right. So unfortunately with conifers, I mean, it's like impossible to climb these things.
Right.
It's impossible to get up there, especially with how remote our site is.
Right.
Some people have used like cherry pickers and things like that and hauled those things out into the field and collected insects at the top. But we think that our sampling method overall, the patterns of biomass are going to be represented by sampling at the bottom. Does that make sense? So like,
Well, you don't have much other choice.
yes, we don't have much other choice, but like the peak in arthropod biomass, we don't have any reason to think that the peak would occur at a different time at the top of the tree versus at the bottom of the tree.
The only thing that occurred might be that it's a little warmer. It might be more bugs.
There's definitely patterns of like within a day, the tops of the trees are the first things to warm. So, but I don't know about like over the whole day or something like that. The other thing is that our poop data, which I now lovingly refer to it as, shows us that we are sampling the right things. So one of the most common diet items were those green beetles, and we find those all over the place with our beet sheet data sampling, for example. Yeah. Yeah.
So suppose it, in the end, was it your measuring mass of the birds?
Oh, yeah. So we take the nestlings out of the nest when they're 16 days old, and we just put them on a scale and we weigh them. Or were you asking about biomass?
So you had biomass, you had brood mass, and you had nestlings.
Yeah, so the nestling mass is different from biomass. Biomass is biomass of the insects. And the way that I calculate that is I measure the lengths in millimeters of each individual specimen of arthropod, of insect. And then I use length weight regressions to calculate the weight based on the length. So these have, what was that?
You're doing an allometry.
Yes. Yep.
Okay. From a climate change perspective, we know that these dryland environments are increasing in temperature and it's increasing in aridity. So when you're talking about this forecasting, what do you foresee for the chickadees?
Yeah, well, that's a good point for our low elevation site, because we definitely, so we don't expect there to be an effect of aridity necessarily at our high elevation site, because there is always snow there annually. Unless the snow were to melt a lot more quickly and they had really, really, really low snow years up there.
And that's kind of what you expect.
It potentially is. Now, with the Sierra Nevada region, the precipitation overall is projected to increase with climate change. However, the temperature is also expected to increase. And so our high elevation site is at an elevation where we don't know what's going to happen. Either it's going to be too warm and all that snow is going to turn to water, or it's going to be just cold enough to where there's a ton more snow occurring at our high elevation site. So that there's two different scenarios that could happen up there. At our low elevation site below the snow line, there may be more precipitation, but it's all going to fall as rain. And so our low elevation site we think is more susceptible to drought. And so if there are multiple drought years in a row that affect the insect abundance at low elevation, we do expect that this would affect our birds in a negative way. Does that answer your question? Okay. Yeah.
I'm interested in that mismatch you saw between the period of nesting
Mm-hmm.
and the period of peak insect abundance.
Yeah.
That the nesting is occurring basically even before the peak insect abundance.
Yeah.
I'm wondering if that might be some sort of strategy for allowing the nestlings, when they fledge, to now have a better chance to survive because there are more insects out there now
Yeah.
than if they fledged earlier in the year.
Yeah. No. So that's an amazing hypothesis. That is, that's one hypothesis. Another hypothesis is that, and this is sort of probably along the same lines, but in related species of birds, it's been found that birds that are raised earlier are more likely to be recruited into the population. So that might be related to them being able to survive that fledging stage. That might be related to the birds that mature fastest are like more dominant in social hierarchies or something like that. I don't know, but there's something going on with birds being pushed to breed as early as possible, but not too early to where there's not enough food. And we think that this might be especially important with, you know, our birds that are year-round residents of these really harsh environments, and maybe social position really, really matters. Something like that. Yeah.
Do you have any mortality data on the fledglings?
Any more what?
Mortality data of the fledglings? What's the percent that survive?
Oh, so unfortunately, we have really, really low like return of our juvenile birds in our system, like extremely low. Like we will see, I don't know how many.
It's 5%.
About 5% of the birds that we silver band in the nest will actually see back in the population later. So that makes it really, really hard for us to measure things like juvenile recruitment and things like that. However, now that we are getting more and more years of data, we can potentially start to look at that in different ways. Maybe not within years, but yeah.
Yeah, I'm wondering, because that, you know, if you've got adults that are around for many years and they've got their own territories, the fledglings have to go elsewhere.
Yeah.
You know, I'm finding chickadees in my yard over in Hidden Valley now. And I never had before, and I keep wondering, okay, did they come from your area? Your parents have forced them out into the valley?
Yeah.
And I've seen chickadees in our dog park over in Hidden Valley. Which
Yeah.
I've never seen before. I get the impression that maybe they're coming down and coming somewhere to try to find territory.
So we don't expect necessarily for juveniles to be moving that far. So for example, between our low and high elevation site, these sites are what, 7 kilometers, separated by 7 kilometers in distance, physical, like, you know, horizontal distance, right? But we've only ever seen maybe one bird show up from low elevation at high elevation ever. So these birds are not traversing elevations. So there might have been like this slow encroachment into the Reno area by mountain chickadees or something due to there being lots of feeders or people planting more trees and there being more habitat or something like that. But we do not expect birds to move necessarily from Sage Hen all the way down here.
Okay, I'll be looking for banded chickadees.
You should. Please do. Please report them. Get some good photos.
I'll just add on to that too.
Yeah.
So, like the 5,000, right, that we banded of those nest boxes, we have like examples of, say, a bird that hatched in this nest box right, when we actually do catch it again with the silver band and we put a capacity integrated transponder on its leg, then that next summer, it will breed in a nest box, sometimes 40 meters away from the one that hatched it. And so the examples of the ones that we do have in regards to dispersal, it's very low dispersal. So in times like there was a eBird article written, I think, a number of years ago, that was claiming that mountain chickadees erupt, and that's absolute garbage. Probably what's happening, so we have absolutely no idea of the composition of the individuals within the birds that we're seeing show up. But what you're probably seeing, especially at this time of year, chickadees are showing up in new places, is you're seeing juveniles disperse. And we know that there is variation in that dispersal distance, but we know it's very, very low. And so the population structure that we have, there is no population structure across the elevation sites around here. So we know that there is incremental, juvenile dispersal. However, that dispersal distance, as far as we know, and as far, and we have banded the most mountain chickadees in the world, right, at this point, and have the longest long-term system mountain chickadees. We know that dispersal distance is low. And so.
Darn, you just ruined my whole night.
Yeah.
New banded chickadees for
Frank and I were referring to them as Ben's chickadee. Ben's chickadee, and they actually did last year, probably my neighbor got it over now. As far as I'm concerned, they're Ben's chickadee.
Yeah.
Well, how do you know your chickadees, the ones, the juveniles that are dispersing, aren't going to the next mountain range? Do you look at the next mountain range?
No, we don't look at the next mountain range. So, I mean, they could be moving within elevations and sort of moving away from where we're monitoring.
Yeah, just to the next.
Yeah.
So there's a whole bunch of Tenemeras that have been there all summer, so I'm not very high, so I don't know, maybe they're yours.
But we don't necessarily have any reason to expect that our chickadee population is like the source population that is like spreading everywhere, right? Because other I mean, there's tons of natural cavities everywhere. So chickadees from the mountain over are also moving over here, right?
You're not seeing them.
Well, we see tons of new birds. We have tons of birds that show up in the fall that are not silver banded, that we did not band as nestlings, and that are first year birds.
And then.
We don't know where they came from.
But are they gonna breed there next summer?
They could. We've seen that, a lot of that. Yeah. Yeah.
Even though they're not feeding their young seeds, are they eating and caching seeds in the summer? Because I keep the seeds in my feed all year.
That's a good question. So in our system, no, they are not eating seeds in the summer. They are only eating insects. Now, urban chickadees are like built different. They have different, they have like evolved a very different strategies for surviving. So they probably cache a lot less. They're probably a lot dumber. Like, to be completely honest, they don't need their spatial cognition when they have food available all year, all winter that's from feeders, right? So you might still have chickadees visiting your feeders in the summer. It's probably just going to be a lot less visiting than in the winter.
What about, I have mountain chickadees breeding down, in the Colin Ranch area, and they've been breeding for years. So, and that's pretty low elevation, and
Yeah.
all those boxes that were there for a while after that study got shut down with the stress, where she was measuring the stress hormones.
Oh.
So Jen, that's Jen Hefner,
Yeah,
and that
right.
study's still going.
It's still going.
Yeah.
So,
Yeah.
so low like that, you get mountain chickadees.
Yeah, oh, totally. Yeah, so I mean, they're coniferous forest specialists, right? And they definitely have like inhabited Reno. And so they're here, absolutely, and they're breeding here. And like I said, they're highly resident, right? And so these birds that we've seen individuals that have bred for 4 years, they breed in the same box, right? Year after year, right? So we know that they're highly resident. And we detect them all winter long too with our feeders, right? As well as just visual observations, and if they come to the feeders to test. And so once they disperse, and they the one thing that we think is going on with mountain chickadees that live in town, that they move a lot more. However, we don't so they have larger territories that they kind of wander around. That's a little anecdotal. We know their flocks are a lot smaller. We know that their densities are a lot less. I will say, so I'm, living in Montana and looking out from the east and looking at all this, this is some of the densest chickadee populations I've ever seen, right? Is the mountain chickadee population in the Sierra Nevada. So we have really high densities. And so the densities in town are lower, right? But they're still pretty high. But they seem to be smaller flocks and they seem to move a lot more. But we really don't know as much about the movement as we'd like. And that's because people haven't made a battery small enough that allows us to put a satellite backpack on a chickadee yet. And so as soon as that happens, we'll actually know how much they are moving.
It's 100% true. I wish we knew what happened to all of our fledglings, but there's no way we're gonna know what happens to them if we can't like have a GPS on them. Yeah.
But there seems to be a bit of an issue of the actual footprint for these populations. Because, I mean, I keep hearing, kind of, a tension between the local populations and what the white population happens to be for the chickens. So I'm wondering, there's a couple different ways to get at this, you know, where you do isotope studies and so on. From there, they get a sense of what the footprint actually is for them. But the other, probably the harder and easiest way is what's the genetic variation within these populations? Is any of that kind of work being done?
There is no population genetic structure. Yeah, so there is none. So there is plenty of gene flow between the populations, even within the city, right? And so we know that they're connected populations. As for isotope studies, that's just a waste of money for chickadees. They don't move, right? So they're highly resonant. And a lot of them, the isotope signatures, the populations, because they don't move and they're not far enough apart, the isotope signatures really wouldn't tell us much anyway, right? And so, because that's definitely something we've thought a lot about. But unfortunately, we just need those tags, right? Because what we're talking about too in movement, you know, they don't fly 10 miles. They're not doing that, right? And so like even that small of movement, they're not doing.
So from a dietary perspective of the isotope studies.
So we're doing fecal metabarcoding for diet, the actual diet composition, right? But actually where their diet is coming from, which isotope, right? Is, you know, there a lot of these animals are living within, you know, like within our system, you know, by the time if they are coming in from a completely different area, like dispersal, we'd be happy to measure those things extremely quickly, right? And so, but they're likely not, you know, given our data, we know that they're not moving very far. So, but we could take better data. So that's true. But like I said, based on all the other biology and the things that we know about mount chickadees, we really wouldn't expect that they're coming from the distance that would be enough to actually detect something in an isotope analysis.
Yeah, so I'm just wondering, you know, birds, in this case, when you're hearing that from the watershed road over right next to you, that there's a possibility of birds coming from there. You know, and then you said there are new birds appearing every year, then, you know, where are they going?
Yeah, well, so that's the, I mean, we have a tremendous amount of natural cavities that are in the system too. And so like, it's all of this surrounding population, right? Is because we only have 350 nest boxes. If anybody wants to help us add, you know, 300 more. So, you know, that's how many we monitor. So we actually find natural chicken cavities 2 feet away.